This recording is going to teach you how to record hematocrit. And what you're going to do is you're going to have 3 or 4 blood samples. This blood sample is in a little tiny beaker. Yours might be in little tiny vials. You need 3 or 4 hematocrit tubes. The hematocrit tubes are small. They have a black line on them, and they say hematocrit tube on the package. If they are longer, they're capillary tubes. Do not use them, please. And we have our centrifuge. Let the teacher turn on and off the centrifuge. So for hematocrit is the ratio of red blood cells to total blood volume. So what we're going to do is we are going to centrifuge our sample and then find out what the volume of red blood cells is and the total blood volume. So first you need to draw a blood sample. So I'm going to just take this blood sample and I am going to put it in. And by capillary action, it is drawing blood up. And ideally, you're supposed to fill it to the black line. We might not be able to fill it to the black line. So now I'm going to put my finger on the top and pull it out. And I have clay plugs here. I'm going to put the clay plug in, and I am going to now put it into my centrifuge. For putting it in the centrifuge, I already have 3 tubes in here. These tubes are across from each other. This tube is by itself, so I'm going to put it across from that tube. So when loading a centrifuge, the tubes always have to be across from another tube. Also, this is the clay plug end. This doesn't have a clay plug. The clay plug end is going here. So this one, it looks like it's already started to settle. Clay plug is at that end. And they are all across from each other. The teacher will then start it. So we are going to now, we have our tubes. We have on the bottom of our tube our red blood cells. So down here's the red blood cells. This part is my plasma to about right there. Then we have a clay plug on the bottom. We're interested in what this volume is, the red blood cell versus the entire plasma volume. There's a couple different ways you could do this. This came with our centrifuge, and you line the 0 up at the top of the clay plug, and you line the 100 up at the top of the plasma. And this sample, because I took so little, is barely at 100%. And I look at what line this is on, and it says 30, 40, 35. It's a little bit below 35. Well, I know the person's anemic since it's below 35, but it doesn't really tell me what their blood hematocrit is. This one, same thing. I have top of the clay plug at the 0, and the plasma at the top, so I move around until went too far. Clay plug is at the 0. Top of the plasma is there, and that is 30 something, and if I look very carefully, I could probably count the lines. I like the old-fashioned way. I have a ruler right here. I am going to put the clay plug at the 0 because all this is a ratio of the red blood cells. So the red blood cells go to here, and the plasma goes up to there. So this is my clay plug. This is my red blood cells. This is my plasma. And the plasma is at about 3.5. So I have, or the total blood volume is 3.5 milliliters. Let's try a different pen. And my red blood cell volume is at about 1.1. So my red blood cell volume from here to here is about 1.1 centimeters. From here to the top of the plasma is 3.4 centimeters. I type that into my calculator. I get 1.1 divided by 3.4, and I get 26 as my hematocrit. So that's a lot lower than this, but it's more accurate because I'm actually measuring the amounts. I'm not relying on little lines. So I'm going to have you guys use rulers instead of these screens because they're not very accurate. So let's do that again. I have this sample. They're definitely anemic. So from here to here is the blood volume. From here to here is the plasma volume. So I line that up. And this person is suffering from severe anemia. So that is my 0 line, my clay plug. That is the top of the red blood cell, which is 0.6. My plasma goes all the way up to 3.8. 3.8. So I do 0.6 divided by 3.8. The 0.6 was my red blood cell volume, the 3.8, the total blood volume from there to there. And 0.6 divided by 3.8, their hematocrit is 0.16. They have severe anemia. And I actually rigged these two so that the people have anemia. So that was expected. And that is how you read a hematocrit tube. Thank you very much.